Okay, good morning. Let's have a class today. We will going to move on to biochemistry test today, and I will split into different sections, and we will do 2 or 3, 2 to 4 each time, and also we're going to interrupt it with some of the clinical microbiology trial, like Urea test, for example, like throat culture. So that we should have fun with this section, okay? Yeah. So we're going to have a lab 11. Today we will do Urea test and 3 cube fermentation test. So first of all, we want to mention about the biochemistry test, very simple. Lecture 3 will talk about the details. So biochemistry test, basically we have a substrate, and the bacteria will have an enzyme to react it and make sure the substrate, the reaction, biochemical reaction can happen in a very efficient way, and then we'll generate products. And the products lots of the time is either acid or gas or alkaline material, and this will cause the color change lots of the time. And then based on this, we can determine what type of the bacteria it is. However, because the biochemistry reaction could be very similar among different bacteria, so how accurate it is roughly about like 80% accuracy. Therefore, in the real lab, we still need to use latex agglutination test. We still need to use molecular methods to further identify it. Including, we already talked about like a gram stem, like a colony morphology, all those things. So just want to give you a hands up what's a biochemistry test. Okay, we're going to do today. First one is Urea test. So what is Urea? The structure is like this. And this is Urea. And the Urea is widely existing in the liver. If bacteria has urease, it's an enzyme, Urease. Now, lots of the time when you see ASE, means this is an enzyme. If bacteria has urease, it will break down, become ammonia and carbon dioxide. Now, how do we know there is ammonia generator? Because ammonia, it is a weak alkaline. So, in this solution, we are adding a pH indicator, which is we mentioned before called phenol red. And the phenol red is very interesting. It's a useful tool in the biochemistry reaction because it adds pH 7, neutral pH, its original color. At acid environments, pH 2 to 3, it is yellow, and at alkaline environments, let's say pH to almost like 12 to 13, it is cherry red. And this reaction is very important for some of the bacteria. Of course, it is harmful. For example, we talk about Helicobacter pylori, HP. That pathogen live in the stomach. And some people have to do a screening test every year to see the HP concentration, because it could end up with stomach cancer. This bacteria has urease. And what happened, you know that at the stomach, the pH is very low. The pH is about 1 to 2. Most of the bacteria will be killed by this acid environment. Now, how HP can still survive? It has urease. this break down urea become ammonia, and this is weak alkali. And what happened? It could buffer the lower pH. Okay, so generally this guy will buffering the pH back to 4 to 6. So that's the environment the bacteria can survive. So that's a urea test. Now today's work is very simple. We have this tube called urea tube, and it should be right here. It looks like cherry red. We will say it's pink. You inoculate the bacteria, and we will do the incubation. And after incubation, we'll see the color change. If it's yellow color, urea negative. If you see sometimes it's generated cherry red, it is urea positive. Okay, so that's not difficult to do. Obviously, you have to label here. Bench number, initial bacteria name, and also the date. Okay? And then you inoculate here. We will prepare you different slants, and it all come from the slant. And this urea tube incubated 35 degrees Celsius, 24 hours. So this is the first thing. Second thing is sweet tube fermentation. Okay, what are the sweet tube fermentation? You will have these sweet tubes everywhere. What are those sweet tubes? Usually you wrap together from here. It will be glucose, lactose, and dextrose. It will be those three. Looks like similar. So we have glucose, dextrose, and lactose. And you have to label them very well. One of them we use a yellow cap, but it goes in order. Okay, they have a bacteria, they have a solution there. So also inside have another tube, looks like this. If you be careful, look see it. So we want to talk about these things. What is fermentation? And we talk about bacteria. During the metabolic process, there is an aerobic respiration, and there is anaerobic respiration, and then we'll come out with another one called fermentation. And they are related to the electron transport or something we'll talk in the lecture. So fermentation, what we will generate? Acid and gas. Okay, how do we know the acid generate? Same thing here. Inside, we add a phenol red. The same thing. This time around, the only difference is we don't care about alkaline. Because acid generates, so what we are looking for? We are looking for more is around here for yellow color. If it's yellow color, acid fermentation positive. How about the gas? This is inside, we have this tube. Got it right here. This is called Duran. It's called Duran tube. The Duran tube we're testing whether there is a gas generated. You can insert to see a very small tube there. How do we know it is gas positive? You see there is a bubble on top after 35 degrees Celsius, 24-hour incubation. So what do you do today? The same thing. Those 3 tubes, you inoculated, you assign the culture. Go here, and go here. What we should not forget? Label on the top. The label will encoding bench number, initial, bacteria name, and also the date. Today, you always remember, this is extremely important. Because otherwise on Thursday, you're going to spending 30 minutes to find your tube. So listen to Rion's order, how you do it. And also these locks very similar. So what we should do, this is also the king cap tube. Don't hold on like this. This is #1 accident in the lab. Very easy to fall off. It's inside another tube. So have your rack to grab all these things. You can have a little bit distance. When you came here, this might be wet. Use your paper towel. Make sure you clean these first, dry these first. Label always on the top, okay? You see the condensation of the liquid? Using paper towel to dry. And then label them on the top. These two looks like very similar. You even need to label what are they, okay? Because you see here, that looks exactly very similar. So make sure you grab it, go back, immediately label which sugar it is. Urea looks different, and then you assign the culture. We have 3 bacteria: E. coli, lactobacillus, and citrobacter, and we will assign the culture. So I start to assign. You only pick assign the culture. The results on Thursday, you share with your bench partner and also share with a small group of 3 of you. Then you can all see the results. Then we'll talk about that. It is not difficult to do. However, you need to do a good job of the inoculation. Make sure there is no cross-contamination. Because otherwise, what will happen? It's all going to look the same. Okay? So, that's all I have, because I have a meeting real quick, and the Rion will take care of it.